Have you ever just stopped to think about the sheer speed of life? I mean, all those countless chemical reactions happening inside you right now, every single second. Well, let's dive into that because there's a wild secret that makes it all work.
Here's the thing that's kind of hard to believe. Under the warm, watery, pretty mild conditions inside our cells, the reactions we need to, live, things like digesting food or even just having a thought, they would normally take hundreds, maybe even thousands of years to happen on their own, right? If biology is supposed to run on a timescale that slow, how are we even here? How can we exist? That is the exact mystery we are going to solve in this explainer. To get to the bottom of this, we first have to understand the main roadblock. It all comes down to an energy barrier that stands in the way of every single chemical reaction in the universe. Okay, think of it like this. For any molecule, let's call it a substrate, to turn into a different molecule, the product, it can't just slide downhill. Nope, it has to be pushed up and over this big energy hill. That very peak is called the transition state. And the energy you need to get up there, that's the activation energy. And the rule is simple. The higher the hill, the slower the reaction.
So life clearly has a massive speed problem, but nature's solution is, well, it's just brilliant. It's incredibly powerful. The solution is enzymes.
Enzymes are these huge protein molecules that act as nature's catalysts. And their key feature is this special little pocket, you can see it right there, called the active site. This is where the magic happens. A substrate molecule just pops into this pocket, and all of a sudden, a reaction that would take centuries is over in a flash. So, just so we're all on the same page, let's meet the cast of characters. We've got the enzyme, that's our catalyst doing all the work. We've got the substrate, that's the molecule it's working on. And then we have the active site. That's the stage where this whole chemical drama unfolds. So how do they do it? This visual really gets to the heart of their strategy. See, the enzyme doesn't change where the reaction starts or where it ends. Instead, it creates a completely new path. That's the blue line. It's like carving a tunnel through the mountain instead of climbing over it. By lowering that activation energy hill so dramatically, it lets the reaction just race to the finish line. Now, for more than 100 years, scientists have been trying to figure out exactly how enzymes manage to lower that hill. And the first big idea was beautifully simple, it was super intuitive, and as it turns out, it wasn't quite right. This idea was called the lock and key model, and it's just what it sounds like. The active site of the enzyme is a perfect, rigid match for the shape of its substrate, just like a specific key fits perfectly into its lock. And this whole concept came from the brilliant chemist Emil Fischer way back in 1894. And for decades, this idea totally shaped how we thought about biochemistry. I mean, it makes perfect sense, right? A perfect fit creates perfect specificity. But here comes the twist. What if binding that substrate perfectly is the absolute worst thing an enzyme could do? What if that perfect fit doesn't speed the reaction up, but actually stops it dead in its tracks? And this question leads us to the real secret of how enzymes work. It's a way more subtle idea, and honestly, it's so much more brilliant. To really get our heads around this, let's imagine a fake enzyme. We'll call it stickase. Its one and only job is to take a little middle stick and break it in half. Now, to break the stick, you first have to bend it, right? That bent shape, that's our transition state. Okay, so if we build our stickase enzyme using that old lock and key model, its active site would be a perfect fit for the straight, unbroken stick. But look what happens. By binding that straight stick so perfectly, the enzyme is actually holding it in place, making it harder to bend. It's stabilizing the very thing it's supposed to change. This enzyme is terrible at its job. So this is the crucial insight. This is the whole secret. A truly effective enzyme isn't shaped like the starting material. It's shaped to be a perfect complementary match for the unstable, high-energy transition state, the bent stick. The enzyme binds most tightly to that fleeting, hard-to-reach shape. And by grabbing onto and stabilizing the transition state, it makes getting there so much easier. So let's put a proper name to that bent stick moment, the transition state. It's that fleeting, unstable, high-energy point right at the peak of the energy hill. It's not the substrate anymore, but it's not the product yet. It's that in-between becoming phase. And that is precisely what a great enzyme is built to grab onto. Okay, so the enzyme stabilizes the transition state. But wait a minute, where does the energy to actually do that, the energy required to lower that massive activation hill, where does it actually come from? It almost seems like we're getting something for nothing here. But, in biochemistry, there is no free lunch. That energy has to come from somewhere. And the answer is something called binding energy. You see, as the substrate bends and strains into that transition state shape, it suddenly forms a whole bunch of new, weak connections with the enzyme. Things like hydrogen bonds and ionic interactions. Each one of those little bonds releases a tiny puff of energy. And when you add all that energy up, it's what pays to lower that big activation energy barrier. Here's another way to think about it. When the substrate first enters the active site, the fit isn't great. It's a little clumsy. But as the reaction starts and the substrate contorts towards the transition state, the fit becomes perfect. That's the moment when all those weak bonds click into place, releasing all that binding energy that powers the whole catalytic process. And the effect of this is just staggering. Take this one enzyme, orotidine monophosphate decarboxylase. It speeds up its reaction by a factor of 10 to the 17th power. To put that in perspective, that's the difference between 1 second and 3 billion years. That is the incredible power of stabilizing the transition state. It's just an incredibly sophisticated, elegant solution that life evolved to solve its fundamental speed problem. Which really leaves us with one final, fascinating question to think about. Now that we understand this incredible secret, what happens when we start designing brand new enzymes to solve our problems?